right, that was an abrupt end. My name is Jesse, and I am with Exploring by the Seat of Your Pants. Thank you so much for being here today. I know we've got a lot of new classes today, joining both live and on YouTube and Facebook. So I want to say that we are all about bringing conservation, adventure, and science into classrooms around the world with free live digital broadcasts. So we do 50 plus of these a month, featuring amazing scientists and explorers from all across the globe. And if you're new to this, I hope you get a chance to join us live or on YouTube today and in the weeks and months to come. So today marks the beginning of a very special partnership, one that we started back and we wanted to begin in March, and then COVID got in the way, and so we're diving in headlong with today, and that is with the Gairdner Foundation. So the Gairdner Foundation is based in Canada, and they celebrate the top biomedical and global health researchers around the globe with a series of annual incredible prizes highlighting their work. I think over a third of all the prize winners they've ever announced at the Gairdner Foundation have gone on to win Nobel Prizes. So they have a good track record of picking some pretty incredible people doing some pretty world-changing work. So today's talk is very outside of the realm of our everyday here at Exploring by the Seat of Your Pants, and that's what makes it all the more exciting. So it's been a slow news week. I mean, there hasn't been much going on, but maybe you guys picked up today that Pfizer, one of the big companies in the world for producing vaccines, announced a really exciting development in the search for a COVID vaccine. And so, of course, we've been tracking this for many months now. We're all excited about the prospect of getting back to normal in our everyday lives. And that is what our speaker today is going to speak about. Dr. Tania Watts is an immunologist at the University of Toronto. Her work has touched upon infectious diseases all over the world, and in finding vaccines and tools to help prevent them and help vaccinate the public and promote public health generally. So I'm really excited to hear from her, talk about her own career path, how her work is in general around the world, and dive in with COVID vaccines and a little bit about today's development as well. So without further ado, thank you so much for joining us, Dr. Watts, and take us away.
All right. Thank you, Jesse. Thank you very much for the invitation, and to the Gairdner Foundation for introducing me to this great organization. And I'm really happy to be here today with all the students and to talk about my favorite topic, immunity. So what I thought I'd do today is tell you a little bit about pandemics and how our current pandemic fits in, and then how we mount an immune response to viruses, how we remember that immune response, and that's the basis for vaccines, and then what are the new vaccines that are being developed. And it's a very exciting time because science has moved so fast on this vaccine. So I'm going to delve right in. We're talking about the disease COVID-19. It's caused by a virus, SARS-CoV-2. So you'll hear me say SARS-CoV-2 throughout. And it's a coronavirus. And coronavirus is a name for their crown-like appearance that you see in this electron micrograph. And these spikes that they have form the crown, and this is how they attach to cells. So human coronaviruses have been identified initially in around the 1960s, and there have been 4 seasonal coronaviruses that give you respiratory, like mild colds in the fall and winter season, but they haven't been very severe, so they didn't get much attention. However, back in 2002, a new severe acute respiratory syndrome emerged in China. And this was initially thought to come from exotic animals, but the sequence showed it was similar to a bat virus, a coronavirus. And it eventually spread to 26 countries, but only caused about 8,000 cases because it's not that easily spread between people, but about 10% of those people died. So it was very severe. Then another coronavirus jumped to humans in 2012, first noticed in Saudi Arabia and thought to come from a camel coronavirus. Again, it spread to several countries, caused a few thousand cases, and a very high death rate, almost 30%. But again, it didn't really cause a major pandemic because it didn't spread easily between people. And now, unfortunately, we've had a new coronavirus emerge in late 2019 in China. And again, the sequence looks like it's related to the original SARS, although a little bit different, and also to a bat coronavirus. And unfortunately, this has now caused more than 48 million cases, more than 1.2 million deaths. And although some people have very mild infection, a substantial proportion still have severe disease or death. And all of these are examples of what's called a zoonosis, a disease that jumps from animals to humans. So scientists have been worried for a long time about a new pandemic coming. And the reason is a very famous zoonosis was the introduction of influenza virus into humans. Influenza actually exists mainly in wild ducks and geese in the wild, and every now and again, a new virus jumps into humans. And the most famous pandemic of influenza was the Great Flu Pandemic of 1918. It came in 3 waves, starting in the spring of 1918, just at the end of World War I, and 2 more waves were the most fatal. And by the end of it, 1/3 of the global population had been infected, and about 50 million had died, with a death rate of 2.5% approximately, which is about 25 times higher than normal seasonal influenza that we have every year. So at that time, we didn't know anything about the virus. They called it influenza, but the actual virus that caused it wasn't identified till 1934. And the actual sequence of the virus wasn't obtained till the 1990s, when scientists were able to reconstruct it from information gained from digging up bodies in the permafrost in Alaska. And you can actually read that story. It's really interesting if you want to know more, in a very nice book by New York Times science reporter Gina Kolata, Flu, the story of the great influenza pandemic of 1918. But so severe was it that as if you look at life expectancy over the previous century, it dropped by 10 years during that year of the Spanish flu. It was called Spanish flu because it was noted in Spain, but we don't know that it originated there. It was so prevalent that the children at the time had a nursery rhyme. I had a little bird, its name was Enza. I opened the window and influenza. So how does that compare with our current pandemic? So this is from the WHO in June 2009, when we had a flu pandemic. We've had a number over the decades since the 1918 flu, and the most recent was in 2009. And you can look at this on a scale of how severe the disease is from low to high, and how easily it's transmitted from low to high. So the great 1918 pandemic was very severe because it transmitted easily between people and caused severe disease and a death rate of about 2.5%, compared to seasonal flu, which is easily transmitted, but a lower death rate and affecting the very young and the very old predominantly. The original SARS had a very high death rate, but wasn't easily transmitted between people. And similarly, avian flu that first killed someone in Hong Kong in 1997 has caused sporadic outbreaks, but doesn't transmit well between people. But what about our current pandemic, SARS-CoV-2? Well, we don't really know what % of people actually die yet because it's hard to get the denominator. But of confirmed cases, the WHO reports a death rate of 2.5%. And we know it's easily transmitted between people. So it's definitely much worse than our regular seasonal flu. So what does a coronavirus look like? The coronavirus is an envelope virus, and I told you about those spikes. They're on the surface, and they're used to attach to cells. Inside this envelope, it has a single strand of a genetic material, a ribonucleic acid or RNA, that encodes all its structural genes, as well as some non-structural genes, such as an enzyme, a polymerase, that it needs to copy itself. The coronavirus also has genes encoded that help it evade the immune response. We'll talk more about that in a second. Compare that to influenza, a different kind of virus, but also an envelope virus with a spike called a hemagglutinin that attaches to a different receptor than SARS-CoV-2 and carrying 8 different genetic segments of RNA. And that's an interesting difference in that sometimes we get new strains of influenza, like in 2009, when 2 different influenza strains mix in an intermediate species such as the pig, and you get reassortment in a whole new virus that can expose the humans to a virus version that they hadn't seen. In addition, influenza has a very high mutation rate. Its polymerase is very sloppy. So sometimes we get a vaccine at the beginning of the flu season, and by the end of the flu season, that virus might have escaped by mutation. One good thing about the coronavirus that makes it more promising for vaccines is it only has the one RNA segment, so we shouldn't get reassortment with different coronaviruses. And also, its polymerase is a little bit less sloppy than the one for influenza. So although scientists have detected mutations, there's not so many, and they don't occur so fast. So there's a hope that we don't have to keep reformulating vaccines for this virus like we do for flu every year. So how are influenza and SARS-CoV-2 spread? Well, they're both respiratory viruses. So here's a picture shining a light on someone sneezing, and you can see the spray of droplets that come out of their mouth. So it seems that the main route of transmission of these respiratory viruses is when droplets land in your nose, mouth, or throat, and they attach to the airway epithelial cells and start replicating. You can also get infected if somebody sneezed on a surface, you touched it, and then touched your mucosal surfaces. But we think the main route is through these droplets. And whether they're droplets or smaller aerosols is a bit of a debate of how small a particle transmits. So how does SARS-CoV-2 infect cells? So now you've had these droplets land in your airways, and some of the viruses will attach to your airway epithelial cells. And SARS-CoV-2, that causes COVID-19, attaches through its spike to a receptor called ACE2. That's a different receptor than used, say, by influenza virus. And it uses this receptor to gain entry into the cells, where it then hijacks the host machinery to make many copies of its RNA and many copies of all the proteins it needs to make intact viruses. And then millions of progeny get produced and can infect neighboring cells, so that you get a cascade of infection that spreads through your airway cells. So in the meantime, within hours of this initial infection, the innate immune system is poised and ready to recognize an intruder and send out alarm signals. Almost all the cells in our body have the capacity to recognize a viral infection and produce an important set of molecules called interferons that interfere with the viral response by causing an antiviral state in the neighboring cells. However, viruses that are successful in humans have to try to counteract these measures, so most viruses also have genes that interfere with the interferon response. So there's this battle in the first 2 or 3 days of that infection, while the immune system tries to limit the amount of virus and the virus is trying to escape and replicate. Other immune cells under your airways are also sounding the alarm and releasing molecules called cytokines that communicate and bring other cells of the immune system in to fight the infection. But if you need to eliminate the infection, you actually need your adaptive immune system. The adaptive immune system consists of 2 kinds of white blood cells called T cells and B cells. And they're really needed to recognize the infection and get rid of it. And as you'll hear, they're also going to be responsible for remembering the infection. So after the virus gets into your airways and starts replicating, some cells of the immune system called dendritic cells pick up virus particles and move them to the lymph node. And so if you've been really sick, you might feel those swollen lymph nodes in your neck. That's your immune system at work. Because when the cells, the lymphocytes in the lymph nodes, recognize a specific viral infection, they start dividing and making more copies of themselves. So the T cells come in 2 flavors. A CD8 or cytotoxic or killer T cell that's actually going to go back out to the site of infection and eliminate the virus factories. Another kind of T cell, the helper T cell, helps the B cell to become an antibody producing cell. So inside your lymph nodes are B cells that have randomly arranged receptors, a different one on each B cell. And if one of those B cells has the right specificity to bind that new virus, it will start dividing and with help from T cells become an antibody producing cell that will go out into the circulation and provide a supply of antibodies circulating through your body. Some of these cells can go and live in the bone marrow for decades, and some infections give rise to lifelong immunity, although not all infections do. And some of the seasonal coronaviruses, we don't think you get such long-lived immunity. So now the T cells that we've developed in the lymph nodes have gone back to the site of infection, your airways or your lung, and they find the infected cells that have essentially virus factories, and they can kill those cells. So although killing the cells might be a bit of collateral damage, it's very important to eliminate the virus factory so it doesn't keep producing more virus. And the antibodies are also very important. Antibodies can bind to the virus and cause it to be captured and eliminated by cells of the immune system. And there's also a very important class of antibodies that we're very interested in for vaccines, and that is called a neutralizing antibody. That binds to the spike protein in such a way that it blocks its attachment site so it can no longer get into cells. So the combined effect of the T cells and the antibodies is to eliminate virally infected cells and to get rid of the free-floating virus. So now you can completely recover from the virus infection. So we've eliminated the virus, but how do we remember that we've been infected with that virus? What stops us from being reinfected? And you may have heard that about 40 million or so people have had the coronavirus. So far, we've only heard of a couple of reinfections. So most people are immune and protected, at least in the short term, from reinfection. So this is a diagram which shows how many T cells or B cells you have over the time of infection. So imagine this is the lymph node, and you have millions of different T cells. And let's say this one T cell, the purple T cell, is specific for this new virus. It recognizes it, and it starts dividing and contributes to the elimination of the infection. After you've eliminated the infection, we don't need all those T cells. You don't want swollen lymph nodes indefinitely. So many of those cells die off. But a small proportion persist in the circulation and in the tissues as memory cells. And these are in higher number than you started with. So if you get re-exposed to the same infection, you have more of these to start with, and they're actually poised to respond more quickly to an infection they've seen before. So this is shown another way. Many scientists throughout the world have been avidly studying the immune response to SARS-CoV-2, and they've shown, in fact, that after people have cleared the infection, they have T cell responses that spike initially, and although they decline somewhat, we've already detected these T cell responses several months out, work from a number of labs. And also memory B cells persist. And we also have circulating antibodies that, although again they decline a bit over time, you have much more of them than you started with. So this is the basis of how we're protected from reinfection, but is also the science behind how vaccines work. So to make a vaccine, we want to introduce an inactivated virus or part of the virus into the immune system, into your body in such a way that it will trick the immune system into making T cells, B cells, and antibodies against the virus antigens, the part of the virus that the immune system sees, but in such a way that you're not exposed to any live virus, you're not going to get any risk of infection, but you get protected by the vaccine as if you'd previously been exposed. So a really good place to track this, the WHO has a site, and I particularly like the site the New York Times has called Vaccine Tracker, and it's been tracking all the different vaccines that are under production around the world, or under development. And there's hundreds of these at early stage, and a number of them are already at advanced stages. So let's first tell you about the stages of vaccine development, and then I'll tell you a bit about some of the vaccine candidates. So before you can even make a vaccine, you have to do preclinical studies. So very quickly, scientists identified the SARS-CoV-2 as the causative agent of COVID-19. They identified that the spike protein was how it attached, and they identified that you could make an immune response to the virus by putting in various test vaccine candidates into animals. So these preclinical studies look to see if you can get an immune response to your vaccine candidate and whether it's safe. Once you reach that stage, you then start going into clinical trials. That's a trial where you introduce to human. And the first time a new vaccine is introduced into humans, you have to proceed very cautiously. So the first SARS-CoV-2 vaccine trials involved maybe 40, 45 people, and you introduce increasing doses of your test vaccine, and you make sure it doesn't cause any adverse effects, and you ask if it induces the desired immune response. Does it make antibodies, particularly neutralizing antibodies, and also T cell responses? Some of the trials were done phase 1, phase 2 together. Phase 2 involves a few more patients, maybe in the hundreds, or volunteers, I should say, rather than patients, healthy people who volunteer to be tested. And usually in phase 2, you expand from sort of 20 to 50-year-olds to adding some children, some younger people, and some older people. Because older people, with age, our immune system becomes less effective, and we may not make a stronger response to the infection or the vaccine. Once we reach this phase, then the really critical phase is phase 3 trials, in which we actually ask if the vaccine protects against infection. And so in this case, for the several groups are now in phase 3 for the COVID-19 vaccine, and about 15,000 people would be given the test vaccine, and an equal number would be given a placebo, something maybe the media in which the vaccine is given, or another harmless, already approved vaccine against something else. And then you have to wait as these people go back into the community, and you wait till a certain number of people get infected. And until then, neither the person who volunteered for the trial nor the scientists running the trial know who got what. Only after you decode, after a number of infections, do you find out if the vaccine is protective. And the whole time you're constantly monitoring and asking people to report any events. Most vaccines cause some mild soreness at the site and sometimes even a transient fever because that shows they're inducing an immune response. But you're really looking for anything that looks like a bad response. So as we'll hear today, Pfizer actually decoded some of their samples after 64 infections. And we're really, the whole world is excited to hear that of the people that had been infected, the people who had the vaccine were 90% protected to the people who didn't. And they're still continuing to monitor their phase 3 trial. And I'll show you that there's a number of phase 3 trials going on. Now, once the phase 3 trials are complete and the vaccine is proved to be safe, and that has to be a very high standard because you give vaccines to healthy people, and also that it protects a significant number, say people are looking for at least 50% protection, then the health authorities in each country review the data and decide whether to approve it. Once that vaccine is approved, it's going to be given to millions of people, and you have to keep monitoring for safety in case there's rare side effects once you get to millions of people. So what are the some of the candidate vaccines that are being developed for SARS-CoV-2? So again, remember, we've got to give the vaccine in a way that makes the immune system think it's seeing the virus, but is completely safe. So one of the simplest kind of vaccines is a whole inactivated vaccine, where you actually inactivate the vaccines, usually somehow chemically, so that the virus can't replicate. But it looks like a virus in order to induce an immune response. And there's a couple of Chinese pharmaceutical companies, Sinovac and Sinopharm, that have already had approval for emergency use in China, and they're still undergoing phase 3 trials for these vaccines. Another way that's been used by several pharmaceutical companies for SARS-CoV-2 is to take another common cold virus called an adenovirus, and to take away some of the genes that are essential for that virus to replicate in cells so it cannot infect, and to replace those genes with just the one gene encoding the S, or the spike, of the coronavirus, with the idea of inducing an immune response to the spike. And so this has been done with several adenoviruses. The common cold virus, adenovirus 5, there's some concern with that because many of us are immune to adenovirus 5, so we might block the vaccine and have to use a higher dose. Another group is Johnson & Johnson is using a rarer version of adenovirus that affects human, 26. And the Oxford group, collaborating with the pharmaceutical company AstraZeneca, is actually using a chimpanzee adenovirus. In all cases, these viruses cannot infect you. They can go into the cell once, allow the S protein to be produced, but they can't cause an infection. And these are quite good because they may only require one dose and are quite stable. There's a new kind of vaccine being developed, and this is the one that Pfizer's announced results about today. And in the past, this kind of vaccine has only been tested in limited trials for cancer vaccines. But this involves taking the RNA for the spike protein, and then because RNA is kind of a delicate molecule that's easily degraded, they encapsulate it in lipid nanoparticles, and that also helps the immune system recognize this as something particulate that looks more virus-like. And then again, the cells will take up this vaccine and produce S protein and induce an immune response. Now, the good thing about this one is that it's really quite cheap to produce. While a whole virus requires you to actually grow the live coronavirus, which requires biocontainment, the recombinant adenovirus doesn't require the live coronavirus, so safer to generate, but does require cells and virus production, inactivated virus production. The RNA can be chemically synthesized, so that's going to be able to be mass-produced quite well. One caveat is it needs to be kept cold, and I think currently they're thinking of dry ice shipments, and we don't know exactly how stable this vaccine will be over time. So that may limit it in settings where you can't keep the virus cold while you deliver it. There's another kind of vaccine under development that's called a subunit vaccine, where you just already purify the S protein. That takes a bit more work than making the RNA, but can be a very potent vaccine. And they combine this with an adjuvant. An adjuvant is an immune-enhancing substance really given to make something as simple as the S protein trick the immune system into think there's an immune response and cause inflammation and call in the troops to fight the vaccine, which isn't an infection. So a number of these vaccines have been in clinical trials now, from phase 1 up to phase 3, and they all induce neutralizing antibody responses as well as T-cell responses. So we're quite excited about that. So if these vaccines are approved, why should you take a vaccine? So there's 2 reasons to take a vaccine, and one is to protect yourself, and the other reason is to protect those around you. And that is the concept that you may have heard in the news about herd immunity. So imagine that you're one of these blue people here that haven't been immunized but is still healthy, and into your midst comes a couple of coronavirus infected people, and nobody's immune, and the virus rapidly spreads. If the vaccine was available to only a few people, and the same thing happened, those few people would be protected from infection. But the other people around the infected person would still be exposed, and we'd still have a rapid spread of the infection. If we can get a large number of people immunized, then if the coronavirus infected people come into their midst, this can stop the spread of infection, because viruses don't replicate outside a host. They can only infect someone else if that person gets exposed before the immune system of the person carrying the virus completely eliminates it. So if you have enough vaccinated people, you can cut that transmission. And this is very important, because these few people here might be people who, through some genetic variation, are a rare non-responder. They might be someone who's immunocompromised, they've had a transplant and don't make a good immune response. Or they could be the very old people who, as we age, our immune system becomes weaker, and we're not as good at making a new immune response. The case of flu, we see the very young and the very old susceptible, whereas in the case of COVID-19, it's much more skewed to the very old, with the young being more protected. So vaccination is an important way to achieve immunity without any danger of infection. And we don't know yet how many people need to be vaccinated to break the spread of infection. With some diseases, such as measles, which is one of our most infectious diseases known, when vaccination rates drop below 90 to 95%, you see outbreaks. The estimates I've seen for SARS-CoV-2 is more like 50 to 60%. So that's a very hopeful number if enough people can get vaccinated. So vaccination is really one of the most effective medical interventions devised by man. It's led to the complete eradication of smallpox in the 1970s, led by the WHO. And now there are many effective vaccines against serious childhood diseases such as polio and measles. Several of the COVID-19 vaccines that are in phase 3 clinical trials have been shown to induce strong immune responses. Which of the vaccines will protect? Well, as we mentioned, Pfizer today announced 90% protection when they looked at the first 94 cases. They actually need to get to about 164 to complete their trial. But based on this early result, they have to watch another couple of weeks for safety. They're going to apply for early approval. What we don't know, because these tests are done this is information very soon after the vaccine, is how long this vaccine-induced protection will last. Will it give lifelong immunity, or will we need an annual shot like we do for flu? And that remains to be seen. Another important thing is, when will we have enough vaccine? So Pfizer says they'll have 50 million doses this year and 1.3 billion by 2021. So we really need many of the vaccine candidates to be successful to really cover the whole world. And I think the science is very hopeful. And as I mentioned, we don't know how many to have herd immunity. So I'm going to stop talking now and give you a chance to ask me some questions about what we know about SARS-CoV-2. So thank you for listening.
Thank you for that. That was fantastic, Dr. Watts. And again, we've covered so much, and these are issues that are of so much interest to so many classrooms and communities across Canada, across the US, and around the world. So really appreciate you sharing your time with us today. We've got classes in Ontario, in California, in Pennsylvania, all on YouTube. So if you want to share questions in the YouTube chat bar, please do. Our live classes, I'm going to come to all of you in just a second. But I want to start off with a question about another vaccine, actually. So in Canada, certainly in Toronto, where we both are, the flu vaccine is getting increasingly hard to find. And we're really encouraging as public health professionals to get people to get the flu vaccine. Why is that important?
Well, I think one of the really important things about influenza is every year influenza causes about 500,000 deaths worldwide, even with the regular seasonal influenza. And in Canada, it's about 4,000 to 8,000 cases. And it's going to be difficult for the medical professionals when you first come in with respiratory disease to know whether it's SARS-CoV-2 or influenza. So we don't want to overwhelm the hospitals with influenza, and we really want to make sure we don't cause any confusion there. So I think the more people vaccinated, the better. What we don't know yet is if all our measures, so if we're all wearing masks and social distancing, we hope we can get less flu than usual too this year.
Yeah, fantastic. And this is something that you're hearing from public health professionals. I just really wanted to get a chance to talk about it in today's broadcast. So we're going to go to Belmont, California first to Ms. Gerard's class, joining us in Notre Dame High School. So Ms. Gerard, just demute your microphone.
All right, hello. Thank you, Dr. Watts. I'm here with my students, AP Biology class from Notre Dame, and I have a bunch of really great questions. So I'll start with one, which is, what do you think is a realistic time frame for the widespread availability of the vaccine? And about how long do you think it'll take before we get back to normal in the world?
Yeah, that's a really good question. So just from hearing, Pfizer is just one of the companies, and it's going to take time to produce the vaccine. But the good news is they're not the only vaccine company. There's, 7.7 billion people in the world, and there's going to be a logistical challenge, getting the vaccine everywhere. I think we'll see starting in December, January, some of the countries that have arranged vaccines will be able to vaccinate some of the high-risk people. And then I think the broader population, it's going to be later in 2021. And I think the hope in most countries is to prioritize the older people. One of the good news is so far, several of the vaccine candidates seem to work in older people. So there was a concern whether they'd make a good enough immune response. And I would say they induce an immune response in older people. We don't know yet if they're protected. And and the healthcare workers and frontline workers will be a high priority. So I think it'll be several months still before we start to see the impact of the vaccine, but it's very promising. And the fact that so many companies have stepped up and moved so fast, it's very exciting to actually see a vaccine from discovery of a virus to licensing could have happened in 1 year is really phenomenal.
It's worth, I'm so glad you brought that up. And again, we'll probably touch upon it in some of our later questions, but it really is one of the great medical and scientific accomplishments of all time. The amount of clinicians, scientists, doctors, researchers like yourself that have been involved in the creation of this is absolutely mind-blowing. Like, this will go down in history as a really incredible time, a scary time, a difficult time, but I'm glad that there's so much positive work being done. Awesome. All right, I'm going to go to Ms. Cormier's class, joining us in Dartmouth, Nova Scotia. If you guys want to come up for a question, go for it. Don't be shy, I know. And if you don't, I can come back in a minute. That's okay.
Oh, we got one coming.
Got one coming.
So just Hello.
I had a question. I don't know if it'll be able to be answered, but I know a lot of places they charge a lot for medical stuff. Yeah. So I was wondering if there's projected prices, or if it's going to be tried to have the COVID vaccine to be free and accessed for all.
Yeah, that probably depends on the country. So I think Europe and Canada are likely to pay for the vaccine as part of their public health program. I think the US is having some sort of plan. And of course, there's a major effort by the WHO to make sure that the vaccine doesn't go to only rich countries and to distribute throughout the world. Now, India has a huge vaccine production capability, and they've promised to also make a lot of vaccines and make sure they give some of it to countries around them. So, yeah, it is going to be an issue. Availability, who has to pay? I don't know the exact prices, but I think in countries with public health systems, much of that might be paid for.
Yeah. So October 8th, we have Justin Trudeau in Canada, where we are confirming that for all Canadians, COVID will be vaccine will be free. So that's the Canadian situation. And again, if you're in the United States, it might be different around the world. But this is something where, again, we're eternally hopeful. It should not be something that is pay to play for getting a vaccine that is potentially life-saving. So great question to our student in Ms. Cormier's class. All right, Ms. Amon, I'm coming to you in just a second. I want to share a quick question from YouTube. So this is from Owen in Ms. Brett's class. Do viruses mutate once they've infected someone according to their genetics? So genetic and vaccine-based or virus-based changes.
Right. So I know more about this from influenza, which mutates more rapidly than SARS-CoV-2. So let's say you get infected with influenza and you've got your cells are producing lots of copies of the virus. It does it very fast and sloppily. So if it makes copies of the genes that it needs and it gets enough of them made to make a virus, if it has a mutation that doesn't prevent the virus from being generated and reinfecting another cell, that mutation will stick around. If it's a deleterious mutation, that substrain of the virus will die out. So it's a kind of a selection going on all the time. And if your immune system makes antibodies to a particular part of the virus, and that virus randomly mutates, that virus then, if it's still a functional virus, and that's a random chance, then it can still produce a virus that's now escaped that antibody. So that's how viruses can escape the immune response. And that happens with HIV, that's highly variable, and with influenza. SARS-CoV-2 doesn't seem to be as variable. So we're hoping that that's not going to be a major problem with that. And that other vaccines such as measles, polio, the same vaccine's been around for generations. So you don't have to keep remaking it. Whereas flu, we're constantly having to reformulate the vaccine because of mutation.
Yeah, that was fascinating. There's a lot of different scenarios. And again, you touched upon that in your talk too. Some of the classes on YouTube actually were keen on your slides after the fact. And so for everyone who's watching this session will remain on YouTube indefinitely. So if you want to follow up with anything, catch anything you missed, it'll all be there. Ms. Amon asked a great question. So Ms. Amon is joining us in Ontario, a grade 12 class. So I'm going to bring her in. She asked a great question in chat, but it's better to say it live. So Ms. Amon, if you want to share your question with us, go for it. And, oh, we might be hearing you. We might not.
I can see it on the chat. Do you want me to go ahead?
If we're having a little bit of tech difficulties, I'll share your question in the chat. So yes, how do we know there aren't any side effects that might become present after a longer duration after getting the vaccine? A great question. I knew you were going to get this one.
Right. So, I mean, there are requirements for the clinical trials to watch for a certain amount of time, which is why if you read Pfizer's website, they're not immediately applying based on preliminary data. They actually have to watch a bit longer. But from what we know of the trajectory of the immune response and how long it takes to induce an immune response, we would really expect that the effect of the vaccine in inducing the immune response would happen over a period of weeks, not over years. The memory cells will stick around. But so if there was going to be some strange side effect because, there's something in the vaccine that looks like a self-protein that could then attack and give you an autoimmune disease, that would be what scientists are scared of and looking for. That would be seen, we think, within a few weeks. So we think that that's probably long enough, but even so, it would. One of the problems with vaccines is they're given to healthy people. But if you give a vaccine to millions of people, and then 2 weeks later, somebody could be hit by a car or something could happen, you might say, oh, well, that happened 2 weeks after the vaccine. But you have to actually look and try to find evidence if there's a cause and effect. So it's a tricky area when a side effect is rare. But scientists and vaccine-producing companies, doctors have to send reports of severe adverse events, and then they have to be investigated to try to figure out if there's anything to be afraid of.
I'm really, really glad we got that question. I mean, even among my own friend groups and my family members, the concern is, oh, this has happened so fast. What are the concerns for health down the road? And it's worth noting, as you said, that there are tons of really dedicated people that are on the case that are going to be looking into this and making sure.
Yeah, and I should say the science has gone really fast, but they haven't sped up the normal requirements to have careful phase 1, 2, 3 trials, the normal amount of time to watch for side effects. That's what's being done at absolutely the same way a vaccine that had taken 10 years to make. What the difference is that the companies have actually taken a big risk. They really went into production and moving things forward without waiting as long as they usually do. And I think it's the rate of discovery has been, and the pressure to do something has been unprecedented. But the safety hasn't been shortcutted on these vaccine trials.
And we are all very happy to hear that. And thank you so much. I'm so glad we got a chance to say that. So I'm going to go to the Pizzatelli family in Vancouver in a minute. And then I'm willing to take this a little bit longer than our typical 45 minutes, Dr. Watts. I know you're very busy. So
I'm happy to stay as long as people have questions.
Perfect. Well, we'll take a few more than usual today. So Pizzatelli family, come on in, just demute your mic and you're good to go. Hey, girls.
Hi.
Okay, so I know you mentioned that the COVID virus attaches to I think the ACE2 receptor, and I was wondering if somehow in some ideal world you could get rid of that ACE2 receptor in people, maybe through CRISPR or something like that. Could you eradicate COVID in people?
Well, the problem with that is that ACE2 is a very important receptor. I'm not an expert on ACE2, but it's involved in regulation of things like blood pressure and coagulation. So one of the things, I have a colleague who's an immunologist and a cardiologist, and he thinks COVID's actually in the severe patients almost becomes a heart disease rather than a respiratory disease, because people with have complications due to the blocking of, we think because the virus attaches to ACE2, it might actually impact its normal function. So if we got rid of it, I think we would have serious problems in the patients for lack of its normal function in the body. So that is a problem. And then, could you try to block? I think antibodies to the virus that block attachment might be safer than trying to block the receptor itself, because we might block its normal function.
What a cool technical question, ladies. Thank you so much for that. And by the way, plug of the day on our Gardner series on November 17th, we have Rodolfo Berengu, and he's going to be talking about CRISPR. So if you've heard of CRISPR, maybe you haven't heard of CRISPR and you want to follow up on that topic, a nice follow-up to today's presentation. And again, continuing our really exciting series with the Gardner Foundation. So I'm going to take a couple questions from YouTube really quick, and then we'll dive back in with some live ones and go from there. So on YouTube, this is a cool question from Miss Brett's class, Eloise. How and do different seasons affect the spread?
Okay, well, I can answer that a little bit with respect to what we know about influenza virus. So influenza virus is normally a seasonal virus that comes up in November and lasts till about March. And people have done studies of that in the cool, dry conditions of winter, the droplets we spray actually go further. And the virus is inactivated less quickly than in summer, when the humidity makes the droplets fall out of the air faster. So in a normal flu season, it comes up in the fall, winter, and then ends in the spring. However, when you have a pandemic, all bets are off. You remember the 1918 flu started in the spring, the 2009 flu pandemic started in the spring. And the COVID-19, although it's a respiratory virus, and we would expect seasonal coronaviruses to come in the fall and winter, when you have a pandemic with nobody's immune and it's spreading from people to people, it doesn't seem to follow those rules as well. It seems to overcome those limitations.
I'm really glad we got that question. Thanks, Ms. Brett's class. All right, let's go back to Notre Dame High School. If you guys have another question in California, come on back in and go for it.
All right, we do. We actually have been doing some research about SARS-CoV-2 and the impact it has on individuals with different blood types. And there have been some studies that have been done that says O and B are less impacted than A and AB. Do you know any information about that or have anything else to share about that idea?
I'm not an expert on that. I remember reading about that too. It accounts for about 13% of the severe cases. So I actually think there's going to be several different factors that affect who gets severe cases. And I'm not sure if it's something to do with coagulation and the complications. I'm not really, it's not really my expertise. So, yeah.
But great question, guys. Again, really technical questions coming in now. This is awesome, everyone. So Miss Cormier, I'm going to come to you in just 2 seconds. Miss Amon had a couple great questions in the chat, and I'm sorry, again, your audio is a little iffy today. So one's really quick, and I know there's a quick answer, but I want to make sure we get it across. So Dr. Watts, can a person spread the virus that causes COVID-19 even if they have no symptoms?
Yes, unfortunately, it seems, so when you get infected with a virus, the replication is going on, and you may not get symptoms until you really start mounting an immune response. And so it takes about 2 or 3 days to get to peak viral load. But particularly for SARS-CoV-2, we're hearing that people can actually spread the virus a couple of days before they get symptoms. And some people have really mild symptoms and yet can spread the virus. And I actually heard a talk on the radio from somebody at University of Arizona who was saying that the virus is actually interfering with neuropathic pain, and it may be dampening our ability to notice we're sick, which was a clever way for the virus to make sure it gets spread around if we don't stay home. So,
yeah, fascinating. We're touching upon sort of news agency talks and some of the things that you've, research of your colleagues, and this is a really, fantastic opportunity, guys. All right, I'm going to share another one from Miss Amon, a little bit longer. I wanted to get that one in. So for a vaccine to get approved for public use, does it have to offer a cure for every individual tested or majority of patients/volunteers?
Right, so great question. So there has to be kind of a cost-benefit analysis. So for example, we would only allow mild side effects like a little bit of fever or soreness at the injection site. And so I think the FDA in the US has said they want to see at least 50% protection to approve a vaccine. So it doesn't have to be 100%. And because there may always be some people who don't make a good immune response as others. And we also, I think what we're seeing from what the initial results from Pfizer is, and it's very early, but it seems to protect from infection. But one of the scenarios people discussed is, well, what if it didn't completely protect you from infection, but just made it milder so you didn't end up in the hospital or you didn't die? Would that be enough? And I think in this case, it probably would. But each health authority has to review the data and set their own criteria. And this is done independently from the companies, the pharmaceutical companies. The health authorities are going to do a cost-benefit analysis and decide whether it's worth investing in that vaccine.
You address so many worries and concerns and questions that are being asked. This is great. Dr. Watts, let's go to Ms. Cormier's class, again in Dartmouth, if you guys have another question for us. Come on in.
Hello. So my question was, for the vaccination trials, are the patients that are being trialed, are they like chosen by consent, or are they getting paid for the trials?
That's a good question. I think that, to my knowledge, these are, and I don't know all the details of all the trials, but I think there's been a very large number of volunteers for these trials. I don't know if they pay them for their time. I can tell you, when we do studies of immunity to SARS-CoV-2, we get people who've recovered from SARS-CoV-2 to volunteer to give us blood, and we give them a little bit of money to thank them for their time, but not enough to be coercion, and our ethics board has to approve that. So I think that's generally with any kind of trial or medical intervention where you're asking people to volunteer. You don't want to give so much money that people who are vulnerable would be tempted to be volunteering, but sometimes there's a small amount of money, pay for their taxi fare or their time. So I don't know the details of the current trials.
Well, on the ethical note, I want to stress for students that might know that University of Toronto ethics approval would probably be one of the hardest ethical approvals to get any place in the world. So very little chance of any wrongdoing anywhere like that. Quick question, one we haven't got yet, and I just want to stress it. So with the flu, with the COVID vaccine, can you get the disease from the vaccine?
No. So that's a really critical point. So I hope that when I explain the slides, that there is no way that these vaccines can actually create a virus. So in case of, for example, the Pfizer vaccine, which a similar one is also being produced by Moderna in the US, it's a little segment encoding only the S spike. You can't make a virus from one component. You need many, many gene products. So and also they don't even use a virus in the production. They chemically synthesize that gene segment and mix it with some lipid nanoparticles. There's not even even a finite chance of it. It's absolutely not infectious.
Fantastic. I'm so glad we got a chance to cover that. All right, we're going to take one more question. Pizzatelli family, if you guys have one more, just ding your mic and you're good to go.
All right. wondering if it's right to say that pandemics have been getting closer together, because we had SARS and MERS and COVID and the 2009 pandemic all within the space of around 20 years. So is it that they're getting closer together, or are we just noticing them more now?
Oh, well, that's a very good question. I'm not an epidemiologist who studies these things, but I also, in my lifetime, we've seen so many new diseases, Ebola, West Nile, Zika. We're seeing, and many of these diseases, Ebola comes from bats too, actually, as does the coronavirus. So I think it's that we have a very densely populated world, 7.7 billion people, and we're getting new diseases coming to us from the animal kingdom as we're encroaching on more environments, and we're living closer together in larger and larger cities.
Yeah. There's a lot of really cool public science communication on that topic, Pizzatelli. So I'd encourage you to read Spillover by David Quammen. It's a really incredible book, one of my favorite science books of all time, talking about exactly that. The Viral Storm by Nathan Wolfe is another, and Hot Zone, Richard Preston, is a sort of fictionalized version, but one that led to a lot of really cool research on the topic. So great stuff out there, and great question, guys. This has been a super fascinating and unexpectedly fun, considering we're talking about a global pandemic talk today. So I want to stress a few things before we wrap up today. One, this is the first of our series with the Gairdner Foundation. So check out Gairdner.org to learn about all the amazing work that they've done to promote and highlight incredible people in public health and biomedical research. You can check out Dr. Watts and Gairdner on Twitter at Tania UFT and at Gairdner Awards. So check that out when you're done. And for CRISPR, which we got mentioned today, our next talk with the Gairdner Foundation is on November 17th. So check that out on our website, exploringbytheseat.com. Come on in. We'd love to have you guys as a follow-up. Dr. Watts, before we wrap up today, is there any last message you'd like to share with our students joining live from around the world on YouTube about where they can go to learn more about vaccine development, keep apprised of the latest research, anywhere we can send people to keep the learning going?
Good question. So, yeah, I'm trying to think where we should send them. What I would want to say, though, I think, because I didn't say anything much personal about my research, but it's been really felt very important to be so engaged with the community. So many of our colleagues are working on this together, and we're only doing a very small part of studying immunity. But I encourage all of the students there to think about science as a career. It's a very rewarding career, and I'm really pleased to see so much interest in immunity.
Well, thank you so much, Dr. Watts. And I know it's your first time joining us, so you're not familiar with this, but what we do to end every broadcast, I'm going to bring in all our live classes. So Notre Dame, Ms. Cormier's class, Ms. Amon, and our Pizzatelli family, if you want to join me in saying a big thank you to Dr. Watts for joining us today, you are all now in the broadcast. Thank you so much for joining us.